I'm Charli Davies, and I've just finished a PhD with David Richardson. So in this, so far we know that pathogens can act as a really strong driver of selection, and this is particularly true at the immune genes. However, as we've just heard, most animals also harbour a really complex microbial community or microbiome, and this can comprise of the bacteria, archaea, viruses, and microbial eukaryotes. And these microbes can have deleterious interactions, but also neutral and beneficial interactions with the host. And so they've co-evolved with hosts and play a really fundamental role in the host biology and function. And this is particularly true in the vertebrate gut, which I'm going to concentrate on. So the gut microbiome is influenced by a range of host factors, and one of these is the host genetics. And this can play a role in determining the variation and also the composition of the gut microbiome. So the gut microbiome can drive host evolution either directly and also indirectly by affecting various fitness components and providing the potential for evolutionary adaptation to occur. So it's really, really important to try and understand the evolutionary processes that shape this coevolution. Microbial complexity presents a really unique challenge to the host immune system, as it's required to prevent pathogens from colonizing, but also still allow beneficial microbes to remain. So this brings us to the immune genes, and I'm just going to focus on the MHC here. So various studies involving captive animals have found links between gut microbiome composition and MHC variation. And these have demonstrated that this relationship can have an impact on host physiology and susceptibility to enteric infection. However, in the wild, there is only one study on sticklebacks by Bolnick, which is focused on these MHC-GM associations. He found that MHC class 2 diversity and presence of specific alleles was associated with this variation. Although there have been a few other studies linking MHC class 2 variation with the cutaneous microbiome, no other wild studies have tested the association between gut microbiome and MHC or other host immune genes, including MHC class 1. So this brings us to the Seychelles warbler, and this is an insectivorous passerine that lives in the Seychelles archipelago. So to sort of find out whether immunogenetic variation can shape the gut microbiome in a wild species, we first need to make sure that there actually is MHC variation. So in this species, we do know that MHC class 1 variation is maintained and that it's also associated with differential fitness. However, MHC class 2 in this population is still unclear. We need to know if there's individual variation in the gut microbiome. And if these two are both correct, then we can then go and look at whether there are any associations between them. So for this talk, I'm just going to talk about associations with MHC diversity, which here is just the total number of either class 1 or class 2 alleles, and then also presence, absence of specific alleles that are present in at least 20% of the individuals. And overall, we'd predict that MHC class 2 would have a greater impact as these are expressed extracellularly and are so really likely to be important in the recognition of gut microbes. So we caught birds from Kazan Island over 3 field seasons, and using fecal samples, we sequenced the bacterial 16S RNA gene using amplicon sequencing, and then we assigned bacterial sequencing variants or ASVs. We also used blood samples using a similar method to sequence either the partial MHC class 1 exon 3 or the full MHC class 2 exon 2. So on to the results. So first we found that there were 20 MHC class 1 alleles and 10 of these were at greater than 20% frequency, shown in yellow here. And this confirmed previous work in this population. At MHC class 2, we found 24 alleles. However, only 14 of these were full length. 5 alleles were in more than 20% of individuals, although 2 of these were in pretty much every individual. Going on to the gut microbiome, we do find that there's individual variation, and generally they represent the typical passerine gut microbe profile, which is great. So this means that we can move on to this last question. So are these associated? And here we'll do this just by looking at alpha diversity, so the number of species present in a sample, and also beta diversity, or the composition. So are there compositional differences between samples or individuals? So firstly, we find no association at all between MHC diversity and the gut microbiome variation. We did also include other host variables in all of this. So now looking at presence, absence of specific alleles. For class 1, we do find that the presence of 3 MHC class 1 alleles resulted in reduced Shannon diversity, which is shown here. And these alleles also showed a consistent pattern across the 3 alpha diversity metrics we tested, which I'm not going to go into. Looking at MHC class 2 alleles, we only find presence of 1 allele to also be associated with reduced alpha diversity. However, for this one, this was only true when considering Shannon diversity and not the other diversity metrics. So it's a less robust result. Looking at compositional differences, we find the presence of 4 different MHC class 1 alleles to be associated with compositional differences or shifts. And again, we found no effect of MHC class 2 alleles. However, this only explained quite a low amount of variance, although this isn't that uncommon when looking at wild studies in the gut microbiome, as it is such a complex trait. Following on from this, we also went to see whether any taxa were differently associated with these alleles. And we do find this to be true. So just to show one example here, on the left, you'll see those ASVs that are more abundant when the allele is absent, and on the right, those that are more abundant when the allele is present. So there's a lot happening here, but there are several potential interesting taxa that we can gain some broad insights from. For example, individuals that had ASE UA7 present had a greater abundance of ASEs from the order Lactobacillus. And this is a lactic acid producing bacterial taxon, which is often thought to be a beneficial member of the gut microbiome. In contrast, individuals that didn't have this allele, individuals that had this allele had a reduced abundance of ASVs from the order Bacteroideals. And this is an order that's commonly associated with chronic intestinal inflammation, so it could potentially be detrimental to the host. So in summary, we found that variation has been maintained at MHC class 1 and 2 genes in the Seychelles warbler. We also find individual variation in the gut microbiome. Overall, we find no effect of MHC diversity at all. However, we do find that MHC alleles are associated with decreased alpha diversity, and different MHC class 1 alleles are associated with compositional shifts, which lead to differential abundance of certain taxa. And interestingly, most of the effects we find were with MHC class 1 rather than class 2 alleles. However, there's still quite a lot of questions. And some of the key ones here are we don't know whether the gut microbiome may be directly linked to immune gene variation, or indirectly linked via impacts on host health. Although the result that we found more MHC1 means this is likely to be an indirect association. However, importantly, we don't know whether this association actually equates to functional differences in the host, and in turn, can this lead to differences in fitness between individuals? If it does, then this could help explain previous MHC fitness correlations in this species. But overall, hopefully, this is the first step in answering whether immunogenetic variation can shape the gut microbiome. So if immune genes can regulate fitness through modulation of the microbiome, then the microbiome could also act as a driver of selection on immune genes. And with that, I want to thank everyone, especially my fantastic supervisors, so Dave, Hannah, and Martin, all the funding bodies, and everyone else who's contributed to this project. Thank you.